Hello and welcome to the presentation for the poster, A Comparative Analysis of Phenotypes Derived from Genes or Biomedical Literature in COVID-19. This poster was submitted for MedInfo 21 conference and it's authored by Mrs. Sofia Stinson, Mr. Christopher Hawthorne, and Dr. Guillermo López Campos from the Wellcome Wilson Institute for Experimental Medicine at Queen's University Belfast in the United Kingdom. We would like to start with a brief introduction and rationale for the work that is going to be presented. Nowadays, vast amounts of information are contained in the literature, and this information can be extracted and analyzed using biomedical informatics tools and methods. Using SARS-CoV-2 or COVID-19 as an example, in this area of research, since the virus was firstly discovered in 2019, there has been more than 2,000 new papers published a week. This means that there is a lot of information that is hidden there and that needs automatic methods to exploit it and to analyze it. Together with this, we have to keep in mind that diseases are made of different phenotypes. And this is an important and relevant information if we want to understand what are the mechanisms of the diseases. For example, in COVID-19, which is a disease, we can identify different symptoms or phenotypes such as cough, fever, and others. The objective of this work is to present a biomedical informatics approach for the comparison of phenotypes identified in the literature with phenotypes that are inferred from molecular data from the same literature corpus, using a SARS-CoV-2 COVID-19 as an example. The materials and methods for this submission are divided in two elements. Firstly, the generation of the literature-derived phenotypes. For this purpose, we use a series of R scripts that combine the use of the RISMED package for retrieving the abstracts using the following PubMed query: COVID in the title or abstract, or SARS-CoV-2 in the title or abstract, and symptom or phenotype. Then, using the annotations from the human phenotype ontology, we use the ONASYS package to annotate these abstracts that we retrieve initially and identify a list of phenotypes that can be directly detected from the literature. Secondly, we proceed with a definition of a dataset based on molecularly inferred phenotypes. For this purpose, we use a series of in-house developed R scripts that have been previously published, and we use the annotations contained in PubData on the version that was available for the 20th of November of 2020. And we use the following parameters. We use a slightly modified PubMed query that was looking for COVID-19 as mesh term or SARS-CoV-2 as mesh term. And we applied a pathway analysis to identify human phenotype ontologies, HPO annotations based on an enrichment analysis using an adjusted p-value of 0.05, and the adjustment method is the Benjaminian Hopper. We also use a marker frequency threshold of 10, meaning that each of these markers that we are going to analyze for this enrichment had to appear in at least 10 different papers. Going to the results, in this slide, we can see an overview of all the results generated and the methods that were applied. Initially, we started with the COVID-19, SARS-CoV-2, and symptom or phenotype search. For this purpose, we identified 3,042 different papers, documents. We were able to annotate 640 different human phenotype ontology terms, and then we used these 640 terms for further analysis and comparison with those that we identify from the molecular inference process. The molecular inference process, as it was described previously, used a slightly different starting PubMed query that allow us to identify 64,477 different documents. From those documents, we identified those that contain genes, and we limited the query to identify human genes, and this led to a list of 8,012 different human genes. And then, following an enrichment analysis for different human phenotype ontology terms, we were able to identify 801 different phenotypes. Once we compare this with the results from the literature analysis, we see that there is an overlap of 181 different HPO terms. In this slide, we can see more detail about the results. On the right, we have a Venn diagram where we can see the 181 overlapping phenotypes that were identified by both approaches. That represents approximately the 14% of the overall number of phenotypes identified. And it's important to notice that the molecularly inferred phenotype dataset represent approximately 66%, 2/3 of the total number of phenotypes identified. Across those top 10 common phenotypes, and this list is derived from the 181, we can find terms that are familiar, and that is what we would expect, such as fever, pneumonia, dyspnea, fatigue, pain, headache, diarrhea, hypertension, lymphopenia, and vomiting. All these phenotypes have been described and associated with a SARS-CoV-2 infection. It is important to notice that we have been able also to analyze and to identify the molecular mechanisms that may lead to these phenotypes. As a conclusion, we would like to highlight that we have been able to implement a biomedical informatics approach to successfully extract and bring together human gene and phenotype information. We have been able to identify relevant disease gene, disease phenotype, and gene phenotype interactions in COVID-19. And the approach that we have presented today is easily customizable, and it can be applied for other diseases. Thank you very much for your attention, and if you want to learn more about this work, please do not hesitate contacting Dr. Guillermo López Campos at g.lopezcampos@qub.ac.uk. Thank you.